Great. Pnawn da, good afternoon. My name is Mr. Bryn Davis, and I will be doing a AS Biology revision session on Unit 1.2, Cell Structure and Organisation. A little health warning to start with. My first language is Welsh, so there may be some quite imaginative spellings, and I might have to scratch my head a little bit for the English word, but I'm sure we'll get through it. Okay. So session aims for this presentation are a recap of the subject knowledge for Unit 1.2 cells. All right, so this is going to be a proper revision session, so you need a piece of paper to take notes with. If you have a preferred note-taking method, that's absolutely fine, but if not, I'd like to try and use this method, the Cornell method. Okay, so the Cornell method means that you split your page into three, so a line down this third and a line across the bottom third, like that. So you've got 2/3 of the page here. That's your main notes. Okay, so the main things that are said, the main diagrams, stuff like that. Be efficient, paraphrase. Okay, leave out your connecting words. Try and make it short, like a Twitter post. Then on this side here are the cues and reminders. Okay, so stuff that you basically will remind you of what's on this side. Okay, that's a big help. And then the summary after the lecture, you need to try and summarize everything that's in here into this bit. Okay, now that bit there is the start of your flashcards. Okay, so that I find is an efficient and effective way of note taking. Okay. So just before we dive in, there are lots of resources available. So there's the WJC website with lots of different stuff. The key documents there has got your Maes your syllabus, and lots of other teaching guide. There's past papers, all sorts of resources as well. The WJC also have blended learning, which is basically a walkthrough of the unit. Okay, it's a very good resource. Yeah, absolutely brilliant. I've used lots of stuff from there myself. There are also lots of other websites as well. Okay. The resources is about quality, not quantity. Okay, I'd rather you picked one and one that worked and stuck with it, rather than going mad over lots of different resources. Okay. So here is the overview of the unit from the specification. Okay, so it looks quite a short unit, doesn't it? Yeah, there's only four sentences there. Okay, there's two bits of specified practical work at the end, but it doesn't look a monster, but there is lots of detail to learn here. Okay, this is what I would call quite a learny unit. All right. Excuse me. Okay, so here we'll dive in with some content. Okay, so cell theory. So everything alive is made of cells. Cell is your basic unit of life. Organisms can be unicellular or multicellular, obviously, single cell like an amoeba, multicell like an animal. New cells come from existing cells. Yeah, and specialized cells arise from undifferentiated cells. Okay, so those cells that have the potential to develop into any type of cell. And microscopy has allowed us to understand the ultrastructure of cells. Okay, from quite basic to quite complicated these days. Okay, we have a large level of detail these days. Okay, so basically you need to know the structure and function of the cells. Now I know this isn't like the diagram that you're used to at GCSE. There are lots more things in it, okay? And even the things that are in it that you remember from GCSE, they now have more complex functions, okay? So mitochondria, you probably remember. Okay, the cell membrane you remember, the nucleus you remember, okay, the cytoplasm obviously you remember, okay, but now we have other things as well, okay, so you have your ribosomes, you have your nucleolus, which is not labeled there, okay, so that's a nucleolus. Oh, nucleolus. Okay, you have the Golgi body, which is a flattened series of membranes here. You have your endoplasmic reticulum. Okay, the endoplasmic reticulum is a sequence again of membranes, folds that travel out into the cytoplasm, originating at the nucleus. Okay, some are rough because they have ribosomes in them, rough endoplasmic reticulum. Some are smooth without. Okay. Something that you may think is a bit of a red herring is a vacuole. Okay, so you were taught at GCSE that vacuoles only existed in plant cells, sorry, but in animal cells they are small and non-permanent. Okay, but in plants vacuoles are large and permanent. They don't contain sap like plant cells do. Okay, but they are present. Okay, so this is a eukaryotic cell that is that has membrane-bound organelles. Okay, so all the organelles in it have their own little membrane surrounding them. Okay, which is different to prokaryotic cells. Now, prokaryotic cells are the bacterial cells, the more simple ones. Okay. So if you do your sketch of that in the large part of your Cornell notes page and the labels. Okay. Another eukaryotic cell, again, because it has membrane-bound organelles. Okay, so it has organelles that have got membranes in them. This is a plant cell. Okay, so notice some differences. Okay, so there are lots of similarities. Okay, so you've got your nucleus is the same, your rough endoplasmic reticulum is the same, ribosomes, smooth endoplasmic reticulum. You've got mitochondria exactly the same, the Golgi apparatus exactly the same. Okay, but this time there are the additional organelles that plant cells have. Okay, so you have your chloroplasts. Now you know that from GCSE. Okay, but chloroplasts have parts within them. Okay, you also have your cellulose cell wall. Okay, so made of cellulose, that's quite significant. Okay, you have something called a cytoskeleton. Okay, which helps with the structure of the cell, because plants don't have skeletons. Yeah, so they need structure at a cellular level. And then you have what I mentioned before. You have your vacuole. Okay, your central vacuole that is large and permanent. Okay, and that will shrink or swell depending on the osmotic potential of the cell, and we'll talk about osmotic potential in another video. Okay, so again, you need the structure or the diagram of this on the main part of your Cornell notes, and you need to be able to label it. You need to be able to identify what all the pieces are. Okay, good. Excuse me, one of the big things that students have a problem with is identifying the organelles in different cells. Okay, so you are going to get a picture in your exam paper that you've never seen before. Okay, but you need to be able to identify all of the organelles. Okay, so that can be difficult. So part of that is having a really good understanding of the structure, what the organelles look like, what they do. And another part of that is making sure that you've seen enough diagrams of cells. Okay, so the more past paper questions you do, the more cells you see, the more familiar you will be with what the organelles look like. Okay. So there is a diagram in Gymraeg there. So I hope you will be able to tell me that this cell here is eukaryotic because it has membrane-bound organelles, and that is a plant cell because it contains these things, chloroplasts. This one here, despite having straight edges, this is an animal cell. Okay, and that is also eukaryotic, all these membrane-bound organelles. Okay, so you can see your mitochondria there, okay, with its little cristae on the inside. Yeah, you can see your nucleus and the nucleolus there. Okay, you can see the rough ER there, so it's got the bubbles on it. Okay, you can see the Golgi body there, so it's like in a half moon. Okay, so you need to be able to identify all of these parts on diagrams that you've never seen before. Okay. Good. Okay, so this is a list of functions of the organelles in a eukaryotic cell. Okay, some of them are in plant cells, some of them are in animal cells. Okay, this is what I mean by it's a learny bit. Okay, you need to be able to recite, remember all of these functions at this level, okay, for all of these organelles. So you can no longer say the nucleus is the brain of the cell. Okay, it's a very key stage 3 answer. All right, so contains DNA, contains genetic material, replication occurs, protein synthesis occurs. Okay, those are the types of, that's the level of accuracy and the level of detail that you require for marks at AS level. Okay, so again, this will require all of these, all of this malarkey here, okay, all of it on the large part of your paper, okay, and then you need cues and reminders, okay, so you need something to help remind you of what each of these organelles do. Okay, if you can think of a mnemonic for the organelles and then maybe think of, you know, something funny to connect the structure, to connect the function to the structure, that's really good. Yeah, so either way, all of that needs to be in your head. Okay. Right, next bit. So again, like I've said, you need to recognize these structures. Okay, so this is an electron micrograph, so it's an electron microscope basically of a cell. Okay, now you see A there is a mitochondria. You can see it's in kind of a cylindry shape. Okay, and it's got membranes, it's got cristae, it's got folds on the inside. Okay, now annoyingly, this one is also mitochondria, but this one is cut across. This has been cut through the middle. Okay, so you're looking down the length of it. This mitochondria here, you're looking across the top of it. That one, you're looking down the length of it. Okay, so that's another tricksy thing that the questions try and do. Okay, so B is obviously the nucleus. It's the big fried egg looking thing in the middle of your cell, although not always in the middle. Okay, so that's something to be aware of. You've got the nuclear membrane around the outside, and then you've got your nucleolus in the middle. Okay, again, okay, this is a Golgi body. Okay, so it looks like kind of half circly with your membranes going. Okay, so you need to be able to identify what the different organelles are. Okay. Right, all these organelles work in tandem with each other, okay? So the cell is like a factory, and each part does its job on the production line in order for the cell to function properly, okay? So this described separately, but they are interrelated, okay? So protein synthesis, which you will do in detail in Unit 1.5, is an example of how one process, i.e. protein synthesis, occurs in a cell, but it requires lots of different parts of the cell in order to be completed. Okay, so the example there with those ticks is how protein synthesis occurs. So you need to make sure that you're aware of the steps there. Okay, so again, this is the bit. Okay, so all these bullet points, all these ticks here are the bit that need to go in the large part of your notes. You then need in the cues column, okay, something to remind you. Okay, so whether that's a mnemonic or whether it's just, you know, just remembering. Don't underestimate the value of repeating it. Yeah, if you've done it once and remember it, fine, perfect, you're very lucky. If you need to do it 2, 3, 4 times in order to get it in your head, in order to transfer it from short-term to long-term memory, so be it. Okay, repetition is the mother of learning. So if you need to do it more than once, by all means, do it more than once. Okay. Okay. They enjoy asking you to compare mitochondria and chloroplasts. Okay, they're similar types of they have similar types of structure. Okay, they're membrane-bound, double-membraned organelles. They have folds or membranes inside them as well. Okay, so there are similarities and differences between the both. Okay, so you know, an easy way of doing a comparison is like this for your mitochondria. Okay, so a Venn diagram. So what are the structures are in a mitochondria? What are the features of a mitochondria? What are the features of chloroplast? And are there any that fit them both? Okay. There is something here called the endosymbiotic theory, okay, which is a theory whereby it's proposed that these organelles were initially single-celled organisms, free-living single-celled organisms that got absorbed into prokaryotic cells. Okay, so that's a theory that you need to be aware of. Okay. Good. So this time, instead of your Cornell notes, maybe if you just fill in the Venn diagram. Okay, so you need Venn diagram, mitochondria, chloroplasts, and both of them. Okay, good job. Okay, prokaryotic cells. Okay, so prokaryotic cells are cells that do not have membrane-bound organelles. Okay, so that means all the organelles are basically floating around in a big cytoplasmic soup inside the cell. Okay, so again, here, you know, you need a list or a table. What are all these organelles and what their function is? Okay, you know, that's a table. Again, you know, you need to be learning. All right. A diagram, you know, if you're a visual learner, you can draw the diagram and write the function next to them. Okay. So significant differences here. Bacterial cells or prokaryotic cells have flagellae. Okay, so flagella. Not sure what the plural is in say snag, but that makes the cell motile. Okay, so that whips around and enables the cell to swim like a rotor on a boat. Okay, they also have pili. Okay, because it's a single-celled organism, it doesn't have a nervous system, so these are its equivalent of a nervous system, if you like. Okay, so the little hairs that enable it to sense the environment and interact with its environment as well. Okay, here, yeah, you must be able to draw a simple prokaryotic cell and label each structure correctly. Okay, and this is an electron micrograph of a prokaryotic cell. Okay, so I know it just looks like an old it looks like an old black and white TV screen. I'm showing my age there. Okay, it just looks like a fuzzy mess, doesn't it? Okay, but again, you need to be able to identify parts of these organelles in these electron micrographs. Okay, a matter of that is, you know, you need to make sure that you learn what all of these organelles do correctly, inside out and back to front, and then there's practice, you know, past paper questions identifying all the different types of cell and having experience of seeing those cells. Okay, seeing those pictures. Okay, another comparison here. Okay, you can compare these either in a table or you can compare them again in a Venn diagram if you'd like. Okay, so you know, prokaryotic cells tend to be smaller, eukaryotic cells tend to be larger. Ribosomes are free in the cytoplasm here. Ribosomes are large and bound to the rough endoplasmic reticulum. So you'll sometimes see 70S ribosomes and 80S ribosomes. What that 80S means is there's 80 subunits. Okay, so ribosomes are made out of proteins. There's 80 basically amino acids that make up that protein. Okay, so these are smaller ribosomes, 70S, larger ribosomes, 80S. Okay, membrane-bound organelles, not membrane-bound organelles, DNA in the nucleus, free DNA, no nuclear envelope, plasmids present, no plasmids. Plasmids are a circular form of DNA. Okay, so they're an extra bit of DNA. Cell wall peptidoglycan, cell wall cellulose, and then mitochondria present in eukaryotic cells, mitochondria absent in prokaryotic cells, but they have a mesosome. So a mesosome is a fold on the inner membrane. Okay. Right, one thing students do forget is when you are asked to compare two things, okay, you need to compare, what they call it is like for like. Okay, so if they were to say compare prokaryotic cells and eukaryotic cells, you would need to say prokaryotic cells are small, 1 to 10 microns. Eukaryotic cells are larger, 10 to 100 microns. Yeah. What students tend to do is they will list the features of a prokaryotic cell and then they will list the features of a eukaryotic cell. You need to say prokaryotic cell is this, eukaryotic cell is that. Yeah, prokaryotic cell is this, eukaryotic cell is that. That's the case for all comparing questions. Okay, so all comparing questions in your past paper questions, if it's asked for a comparison, this is this, that is that. Okay, not this is all of these and this is all of these. Yeah, like for like, this is this, that is that. Okay, just a generic top tip there for you. Okay, viruses. If you didn't think we could get any smaller, we can. Viruses are tiny compared to even bacterial cells. Okay, so not quite fitting the cell theory. Okay, they don't have a membrane, cytoplasm, organelles, or chromosomes. Okay, they cannot reproduce outside of a host cell. They're basically genes in a protein coat. Okay, arguably they're not really alive. Okay, but they can cause an awful lot of hassle, as we've learned over the past few years. So they can only reproduce inside a host cell, and they tend to be pathogenic, okay, or they can be pathogenic. Okay, so pathogenic means that they can cause you to be ill. So above there's a bacteriophage and an Ebola virus. Okay, notice the units are in nanometers, not micrometers, so we are talking really tiny. Okay, outside a living cell, a virus exists as an inert viron. Okay, so they basically put themselves into some sort of dormancy when they are outside a host cell, but when they get into a host cell, they will infect the host cell and use the cell's machinery to produce more of themselves. Okay, so they will produce more of their genes and protein codes using the cell's machinery. Okay, until that cell then bursts and then infects nearby cells. Okay. Like I said, viruses are pathogenic. Viruses are so small that even bacteria can catch viruses. Okay, I always think it's quite cute to think of a bacteria with a cold. Okay, tissuing into his little hanky. Anyway, right. This table here, okay, is basically a summary of all of what we've done before. Okay, so again, you know, you don't need to do Cornell notes for this page. This would be more, you know, one large A4 piece of paper with your organelles. So these are the lists of organelles here. So you need to put those in this column down there. You need a diagram or a picture, a description. So say what they are, what shape they are, what they're made out of, what their job is, and then whether they exist in a prokaryotic cell, a eukaryotic cell, an animal cell, or a plant cell. Okay. If you can squish all of that information onto one page, yeah, and then repeat it, so the information goes from your short-term memory to your long-term memory, okay, I'd say about half this unit you've got it nailed. Okay, if you can nail structure and function with this unit, you are well on the way. Okay, so that is something else that you can do there. Okay, right. What I would like to do now is to do a few questions. All right, so these questions I have I don't want to say stolen, borrowed from the WJC blended learning webpage. Okay, so all of these questions are available for you to try elsewhere. Okay, what some students do as well is tell me, oh, sir, I've already done those questions, which is fine, it's laudable, it's very good, it's excellent. However, repetition is the mother of learning. If you do them again, they will be easier. Okay, the more questions you can answer, the better. Okay, so what I'd like to do now is I would like you to, on a separate piece of paper, not your Cornell notes, I would like you to stop the video and then identify what these organelles are. Okay, So this one, kind of cylindery shaped, it's got folds on the inside. That should be a big clue. This one here, a stack of folded membranes in kind of a half circle. Okay, so that, okay, big fried egg looking thing is D. Okay, so that's also, you know, I hope this is ringing bells here, ringing bells, yeah. Again, lots of folded membranes, but these have got bumps on them. Okay, good job. Okay, so write your answers down for that, and then I will show you the answers in a minute. Okay, so again, some more. So here are some more questions. Remember, you can pause the video at any time if I'm going too fast, or you need to for you to write the answers down. Okay, so again here, all right, we've got a organelle here that's got stacked membranes inside it. Okay, so stacks of thing inside. A little clue for you, if that was in an exam paper, it would probably be in colour and it would probably be green. Okay, so you name the structure of the organelle there, and then there are other things for you to name as well. So like I say, being able to identify what these things are in detail is essential, and being able to identify what they are on the diagram also essential. Okay. A nice other easy one there, name the function. What is the job of this, excuse me, name the job of this organelle. So that is another easy one. That's just a matter of remembering, okay? So lots of these are AA1, so assessment objective, sorry, A01, assessment objective 1 questions, okay? So it's recall, okay? Practice is the thing with recall, okay? Right, on to question C. Image below shows different structure which is only found in plants. Okay, good job. So I've got cytoplasm, cell membranes, cell walls. Okay, good job. So I need to name what structure X is. Okay, now remember, if you look at that and go, oh, sir hasn't told us anything about that, I've no idea what that is. Okay, you've been told all of them. Have a best guess. Yeah, if you can't identify what it is, make an educated guess, use some common sense. Yeah, what could it be? All right, and then explain the importance. So again, this is a structure and function question. Okay, so structure and function questions, yeah, they, you need to get the information in your long-term memory and then practice your questions. Okay, these are what I like to call bread and butter marks. Okay, good job. So here are your answers for the said questions. Okay, so A was a mitochondria, B was a Golgi body, C was the rough endoplasmic reticulum, D was the nucleus. Okay, and there are the functions thereof. Okay, so again, mitochondria, the site of aerobic respiration or ATP synthesis, not the powerhouse of the cell as we're used to further down school. Okay. B, excuse me, was a chloroplast. Okay, so again, clue there should have been green. Okay, and there are 4 parts there of the chloroplast labeled. Okay, so you need to go down even to the level of labeling the stuff inside the organelles. Function there was photosynthesis. That is the same whether you're in year 7 or you're in year 12. Okay, plasmodesmata. Okay, so if I go back to that there, yeah, these are the little holes in between cells, aren't they? Okay, the little gaps. So that is the plasmodesmata, and any hole allows the entrance or exit of a substance, doesn't it? Okay, so plasmodesmata would be the answer to that one. Okay, right, same again. This time, okay, I'll talk you through the question, okay, and then the next one I'll just allow you to have a go yourself. Okay, so question 2 here, electron micrograph shows some structures found in plants and animal cells but not in bacterial cells. So instantly, yeah, I know all these are membrane bound organelles. I know they're all eukaryotic. Okay, so I've got A, B, C, D, E, F and G. Okay, so name the type of cell found in bacteria, plants and animals. It's nice and easy. Okay, that's a remembering one. That's the first slide. Okay, Name the organelle. So again, cylinder shaped, folds on the inside. Okay, remember. Name structures A to E. Okay, so this is all naming parts of the organelle that I'm trying desperately not to mention. Okay, the organelle that I've just referred to in A part 2. Okay, so structures G are found in all cells. Name these structures and structure F to which they are attached and state their functions. So again, structure function. Okay, what's E? What's F? What's G? What do they do? Describe the difference in structures G in bacteria and in plants. So again, asking you to describe the difference. So you need to do a like for like here. Bacteria is this, plants and animals this. Bacteria this, plants and animals this. Image below shows several structures found in plant and animal cells. Now, I am willing to admit the photocopying or my snipping of the picture there is a little bit squiffy. It's not great, but you can generally see what you need for the question. Okay, so here, this time they've given you the function. Now they want you to tell us what the structure is, sorry, the name of the structure and what letter it represents. Okay, so that again, just recall, what do these bits do? Okay, so I'll give you a couple of minutes now on that, or you can just stop the video and I will put the answers up just now for you. Okay, so the answers are coming up now. Okay, so there are your answers. Okay, so you can stop the video to mark what you've done. Right, the next question, okay, I'm not going to give you a lead into, okay, I'm just going to let you have a go at the question. Okay, so you can stop the video when I publish, when I press the button and you see the question, and then I'll give you a couple of minutes and then I'll show you the answers. Okay, so here is question 3. So you need to write me your answers on a piece of paper. You can stop the video and I will publish the answers in a second. Okay, answers coming up now. Okay, good job. Right then, last question. Okay, same thing again. Okay, there's lots of structurally functiony stuff here. Okay, for you to learn. If you're having trouble with these questions, okay, I would suggest that you need to go back to the first half of the video, okay, and do your Cornell notes again. Okay, so you need to make sure that the information is completely stored in your long-term memory, okay, and that does take some practice. All right, if you are not having trouble with these questions, excellent, well done. Okay, and I will publish the answers in a second. Okay. Okay, so there are your answers for the previous question. Right, okay, so that was the first part, the bit about cells, okay, so structure and function. Okay, so that was the, I'm going to say the not simple bit, okay, because there's an awful lot to remember, okay, but there's not any difficult concepts. All right, so now we need to talk about cells and their relationship to each other, okay, so types of cell organization, okay. So in the same way that atoms are arranged into molecules, cells are arranged into tissues, and tissues are formed into organs, and organs form systems. Okay, so that is something that you have been aware of at GCSE. Okay, so just here. All right, so we're back on our Cornell notes now. Okay, so you need to again, piece of paper, thirds, and then write your notes in the 2/3, cues in the other bit, and summary across the bottom when you have finished. Okay, so cells adjacent to each other in the embryo often differentiate in the same way to form a tissue. Okay, so a tissue is defined as a group of similar cells working together to perform a particular function. Okay, so that is a bread and butter definition. Okay, so you need to know that. All right. The neat trick is remembering all the different types of tissue. Okay. So you have got cuboidal epithelium tissue, you've got ciliated columnar epithelial tissue. So these are types of epithelial tissue. So epithelium, okay, hope you know that that means a covering or a layer or a membrane type of thing, okay? So not a membrane like a cell membrane, okay? So this is like a skin or a membrane in the body made of cells, okay? So there are three types. There are cuboidal epithelium, which, as the name suggests, are little cubes, like these guys here. There is ciliated epithelial cells, okay? So these have little hairs on top. So I sometimes refer to these as Bart Simpson cells because they've got a Bart Simpson hairdo, okay? Columnar there means that they are columns. Yeah, they're not cubes like these guys here. Okay, they're tall and thin with their Bart Simpson hairdo. Squamous epithelium consists of a flattened layer on a basement membrane. So you're talking about cells that are like this. Okay, they've been squished flat. They're not cuboidal like this. They're flat, and they have a basement membrane. Okay, so they are membranes usually found somewhere where you want diffusion to occur. Okay, so the alveolar walls, so there's a short diffusion distance across them. Okay, good job. You can remember pause the video at any time if I'm going too fast for you. Okay. Right, so there we have all sorts of different types of epithelium cells in Gymraeg. So that's your squamous epithelium there, cuboidal, columnar, okay, and there's different. So there's striated cuboidal there, so different ones, and different layered ones as well. Okay, so lots of different types of epithelial tissue. Okay. Muscle tissue is the next one. So this is a different Cornell notes bit now. Okay, so again, muscle tissue. Muscle tissue, you all know what muscles are. Okay, so parts of the body that that incur movement. Yeah. So skeletal muscle attached to bones. That's really movement ones like your biceps. Okay. Smooth muscle, individual spindle-shaped shells that contract rhythmically but less powerfully. Okay, so they can be in the skin, walls of the blood vessel, digestive tract, things like that. And then cardiac muscle, which is cardiac, obviously found in the heart. Okay, so cardiac muscles are cardiac muscle cells obviously contract rhythmically without any stimulation, external stimulation. Okay, the skeletal muscle needs some sort of external information. There needs to be a message from the brain in order to get them to contract, and therefore you move, whereas cardiac muscle does not require that. Okay, it is myogenic. Okay, so a little top tip here. Again, not all cells differentiate to become specialized cells. Some retain as stem cells. Okay, so stem cells are quite nifty in that they retain the ability to differentiate into any type of cell. Okay, which is really good. They can differentiate into any type of cell, which is quite good. It's not difficult, sorry, it's not possible for them to differentiate into, say, new organs. Okay, so if you, for example, had liver failure, your body wouldn't produce stem cells, or those stem cells wouldn't grow a new liver for you. Okay, but that is quite a young technology, and there may be one day, hopefully, that potential. Okay, good. Right, connective tissue. Okay, so connective tissue. as the name suggests, okay, connects or supports separate types of tissue and organs. All right. So areolar tissue is found under the skin, connects organs and tissues together. Collagen, okay, so collagen is in tendons, yeah, connects muscles to bones. Ligaments, which connects bones to bones. Both of these are slightly elasticy, okay. If you have ever snapped one of these, you'll know it's very painful and they take an awful long time to heal. That's due to a poor blood supply to them. Okay. Adipose tissue. Okay, so this is fat cells found just under the skin. So these cells that have particular vacuoles in them that are filled with fat. Okay. So act as an energy store, a thermal insulator, keep you warm, and to protect your delicate organs like kidneys. Okay. So yeah, there we go. Good job. Right. So there is, yeah, you notice this here, yeah. Blood is also a connective tissue for some reason, okay. And you have a picture there of your red blood cells and your white blood cells, okay. So again, there's lots of information here. A sensible thing to do is to summarize it all into one what's this table. Okay, so the different types of tissues, so you need a definition of what tissue is, you need to know what epithelial tissue is, so a general description, and then you need to know the jobs that these guys do. Okay, same for connective and same for muscular. All right, so that is, again, you should be able to reproduce that table from memory. Okay, so if you do it out once from your notes, okay, and make sure you've filled all of these in, then you should be able to do that from memory, hide that. Yeah, repetition is the mother of learning. Ironic, I'm saying that an awful lot, repeating, repetition is the mother of learning. There we go. Right, so tissues will group together in order to make organs. Okay, so again, definition here, organs comprised of several tissues working together to perform a specific function. Okay, very similar to the definition of a tissue. Okay, so you know, your stomach, your kidney, your eye, all of these are organs because there are lots of different tissues working together in order to perform a function. Okay. Excuse me. Something that sometimes students forget is that plants also have tissues and organs and organ systems. Okay, so exactly there. So it talks about a leaf. A leaf is an organ because it has lots of different types of tissues and it performs a particular function. And then you have organ systems. So again, a group of organs working together with a particular function or role. Okay, so again, this particular function malarkey is raising its head here. Okay. So again, lots of different types of organ systems. You have your cardiac system, which is the heart and the blood vessels. You have the nervous system, which is your brain, your spinal cords and your nerves. Okay, so all of those are systems that work together. Okay, so same thing here, guys. We're going to have a little walk through these questions, okay? And then we're going to do some questions independently before we finish. Okay. Diagram below shows images of cells and tissues found in the human arm. State what is meant by tissue. So there's that definition right there. Brilliant. Okay, remember that. Pull that back. Working together. State what is meant by connective tissue. Okay, so again, that's a definition. What does connective tissue do? And you know, you need a level of language here, so don't just say it joins things together. Yeah. Letters from the diagram, which structures shown are connective tissues? Okay, so there may be more than one, just saying. Okay, so you need to choose a letter there. Explain why the whole of the bicep is an organ and not a tissue. So again, you need to know the difference between an organ and tissues here. Okay, so bicep, obviously it's not just muscle, is it? There are other things involved, aren't there? Okay. So moving on to B, epithelial tissues cover large surfaces of organs adapted to carry different functions, including exchange, movement of fluids and protection. The diagrams below show 3 types of epithelial cell, together with an example of where they are found. For each cell, describe how they are adapted to carry out their function. So how are they good at their job? Okay, so A is in the lining of the ileum, your digestive system. So why have we got the Bart Simpson hair going on here? Okay, alveolar wall, nice and flat. Okay, with a short something something, and then lining of the trachea. Okay, so the lining of your windpipe. Yet again, we've got Bart Simpson going on here with his spiky hair. Okay. Just another little top tip, anywhere in biology where there is folds of anything, yeah, it's in order to increase the surface area to absorb more whatever it is. Okay, so anywhere where there's folds, it's in order to increase the surface area. And then identify structure X, which is found in all epithelial tissues. So that question is at the bottom, the basement of that page. Okay. So in a minute now, if you finish off writing those, I am going to publish the answers. Okay? No, I'm not. I'm going to show you the other half of the question. Okay, sorry about that, guys. So detailed structure of skeletal muscle. Type of muscle tissue is striped or striated in appearance due to the presence of contractile units called sarcomeres. Each sarcomere is approximately 2 microns long and can contract 60% of its original length. Calculate the length of one sarcomere when fully contracted. Okay, so this is a calculate the length of, it's a percentage one. So it's asking basically what is 65% of 2 microns. Okay, there are ways of working that out, but that's what it's asking. Okay, now obviously, if you get an answer that's longer than 2 microns, you've done something wrong there, haven't you? Yeah, you need to swap your numbers on the division bit of the question, of your answer, sorry, okay? So you're looking for 65% of its original length. So 65% 2, it's going to be around 1 point something, isn't it? Okay. Muscle retraction requires energy, provided by the organelles labeled M. Identify these organelles and describe how they provide energy for muscle contraction. So again, that's linking us back to our structure and function bit, what we know previously, isn't it? Okay. Right, I'm fairly sure now I'm going to show you the answers. Okay. So make sure you've got answers written down and then you can correct them against these. Good job. There we go. So these are the answers. So you can stop the video there and mark your own work. Okay. Right. So that's gone a little bit longer than I had anticipated. Sorry, but what we've done here is a few techniques and a bit of practice. There are many different ways of revising, and remember, you need to find one that works effectively and well for you. Okay, and that's not just reading your notes. Okay, that's a good top up, but it's not actually moving information from your short term to your long term memory. Remember that you need to recall information. Okay, so that's again, repetition is the mother of learning. If you do it again and again and again until you've got it in your head, and then you need to apply it to old questions in order to test your knowledge, yeah, in order to make sure that you have understood it. Make sure you work hard. I wish you all the best in your exams, and thank you very much.